Hello, everyone. My name is Lucy. I'm a field application scientist with MaxCyte, and thank you for joining our webinar today. Today's webinar is about Express Ticket for Expression, how you can save time and increase reproducibility with ion channel and GPCR screening using the MaxCyte electroporation platform. Today will take about 45 to 50 minutes, and then after that, we'll have a live Q&A. So please stick around if you have any questions. Today's talk outline is as follows. First, I'll introduce the MaxCyte electroporation system and how you can apply it to your ion channel and GPCR studies. The second part will be a case study about expressing difficult to produce ion channels and using high throughput characterization to study KC and Q2, KC and Q3 ion channels. Lastly, I'll go over some experiments and data that we have about how to optimize MaxCyte electroporation for these application areas. So hopefully you'll learn a lot today about MaxCyte electroporation and how you can apply it. This webinar is targeted towards cell-based assays, specifically ion channels and GPCR studies. And so this field is important to many researchers, including you guys, for discovery, characterization, and screening of specific proteins in a physiological context. For example, screening multiple variants of unknown significance to characterize or measure drug response, to investigate mechanism of action, and also to just develop assays to measure these things in a high throughput fashion. And so some of the struggles with these are overcome by electroporation, which I'll go into detail later. And so transfection to produce the cells either stably or transiently expressing the molecules of interest is often one technique used to study these proteins. And specifically today, we'll be talking about how electroporation for transient expression is a very viable and easy to use method. And so these assays can be really difficult. For example, issues with fragile cell types like neurons, trying to get the stoichiometry correct for different subunits of complex ion channels, for example. And then these transmembrane proteins are just very difficult to express and produce as, for example, just like a protein production, antibody production kind of pipeline. So there's only one way to make them and express them inside the cell and measure the cell while it's alive. As well as there's issues with expression levels drifting over time. For example, when you make a stable cell line, and then after many passages, it just doesn't express the same way, or the protein is slightly different than before. And so I'll go over some of the details of how this works, but overall, MaxCyte electroporation can address all these challenges. For example, we can do what we say any cell type with high efficiency and viability, and I'll show you some data to back that up. We also say that we can do any molecule, including multi-plasmid systems for more complex transmembrane proteins. And we can also do all of this reproducibly and consistently. And so what that, well, I'll show you some data to support all these claims as well. So we'll go a little bit upwards and talk about what is MaxCyte, and so what do we offer. And so we like to say that we not only offer the electroporation system, but also a lot of support and help for your research studies. So as I mentioned before, with electroporation, we can deliver any cargo into any cell type, and we can transfect at different scales. So smaller cell numbers, larger cell numbers, we can do anything from around 15 to 20 microliters, which is about 1 million cells, all the way up to 1 liter, which is 10 billion cells. And anything in between we can do very easily. So the scaling up, scaling down is no problem with us. Another thing that's unique about MaxCyte is the scientific support. We have over 35 field application scientists and in-house R&D scientists only doing electroporation. So they're optimizing protocols for many different application areas, for example, electroporating macrophages or NK cells, and we have scientists specifically working on your application area in ion channels and GPCR assays. Lastly, we like to say that we are a partner, not just something to sell you. So we have not only scientific support, but also support from our engineering team who make the instruments and design those, as well as regulatory support for researchers going into cell therapy. We also have relationships with other technology providers, such as Sofion. And so we collaborate and generate data together because our end goal is to help the customers, you guys, you're the researchers, get good results and be able to not worry about optimizing the nitty-gritty details of the experiment, but just get the science done. And so MaxCyte has been around for over 25 years. Our founding in 1999 actually revolved around cell therapy, way before the cell therapy field took off. And so we were a little bit of ahead of our time. So MaxCyte went into drug discovery for a little bit and starting around 2005. And so that's kind of where we got into the field with editing CHO cells and optimizing our system for antibody production. And spinning off of that is how we kind of applied our technology to ion channel assays and GPCRs because we saw a need and a use for it. So I'll go over some of those details with you shortly in this presentation. Going over our expert platform, which is what we call our electroporation instruments. As you can see here in the image on the left, we have 4 different instruments of different scales and use cases. And so I'll go over the 2 ones that you may be interested in after this presentation. And I just wanted to let you know that we have a wide variety of application areas. For example, like I mentioned before, cell therapy, very big for us. We're involved with a lot of different companies getting allogeneic and autologous therapies and T cells, HSCs, NK cells into the clinic. And so those details, if you're interested, we can share with you later, or you can find out on our website. And of course, we also do cell-based assays, antibody protein production, viral vector production, gene editing, and vaccine development. So there's a lot of application areas for our instrument. So if you're in a facility that needs multiple use cases for the instrument before you can acquire it, MaxCyte is quite versatile. So coming back to cell-based assays and protein production, we have 2 instruments specifically for these fields, the ATX on the middle and the STX on the right side. And so both of them have a touch screen and LED indicators. They do static electroporation, and what we mean by that is just electroporating one cuvette and putting the cells into that cuvette. So with that, we can do up to 3.5 milliliters of cells, which is around 75 million cells, if I remember correctly. So that is a lot of cells already, and already sufficient for many researchers here. And then on the STX, the very right side, is our flow electroporation instrument. And I'll show you what that means later, but it allows you to do up to 100 mils of cells or around 2 billion cells. So if you need the high throughput for whatever reason, whatever application, you can do that on the STX. Now I'll quickly go over the electroporation process, which is quite simple and straightforward. So the first part of this is to get your cells and resuspended in the MaxCyte electroporation buffer. We offer a universal electroporation buffer that works with most cell types, and you can add the DNA, RNA, or protein into this suspension as well, and then mix all 3 of them together. You will put everything into what we call a processing assembly or the electroporation cuvette. You'll close the lid and you'll put it into the instrument, and then on the touch screen, you'll select the protocol and tap the green start button. And then after that, your cells are edited and you can proceed with your research, whether that be cell therapy, producing a cell line, or other research. So you might be wondering what kind of cells we can electroporate with MaxCyte, and so I'll go over some of that. In fact, this is just a small portion of our cells that cell types that we can electroporate, but just to show you visually, we can do many different cell lines, as you can see on the left and middle, as well as many primary cell types, including fibroblasts, neurons, skeletal muscle cells, and even iPSCs. So all of these data here are actually from our customer IX cells, and just they electroporated the cells with mRNA GFP or plasmid GFP. And so this is kind of a larger list of the cell types and cell lines that we have done, including many immortalized mammalian cell types, cancer cell lines, primary cells from many different origins, including from humans, from mice, from rats. We can do stem cells, including iPSCs, ESCs. We can do cell types derived from the blood, obviously for very good for cell therapy, including T cells, HSCs, NK cells. And we can also do insect cell lines. And so we have a lot of experience, but if you have something that we don't have a protocol for, we actually have 10 optimization protocols, specifically so that we can tailor what you need for your unknown cell type in order to electroporate it. And so you might want to see some specific examples for those who work with neurons. This is an example from embryonic rat neurons of 3 different subtypes. And so in this example, we electroporated with plasmid GFP mRNA and seeded them into a 96-well plate. And then you can see in the assay on the right side that the viability was above 90%, even and slightly under 90% with the DNA transfection. And you can see that they got about half of the cells to take in the GFP. And then in the second example, we see human bronchial epithelial cells and rat epithelial cells electroporated with YFP and CFTR, and you can see that they were able to titrate the plasmid as well on the middle graph and also the right side. And so the next part that you might be interested in is what kind of cargo you can load into the cells. So on this next slide, we have a wide variety of cargo, including plasmid DNA, mRNA, SSODNs, ASOs like siRNA or shRNA, as well as combinations of these. For example, prime and base editors are usually delivered as 2 plasmids or an mRNA in a plasmid. We also have an example here of CRISPR-mediated knock-in using CRISPR-Cas9 ribonucleoprotein combined with an SSOD knock-in template. We also have people doing transpose on transposase platforms, often for either like cell therapy or cell line engineering, or libraries. And so we often recommend the mRNA transposase with the plasmid DNA transposon for that. And so I'll show you an example here of just multiple plasmids. And so in this one, for ion channels specifically, there are 3 subunits of a calcium V2.2, as well as an inward rectifier Cur 2.1. So this required simultaneous co-electroporation of multiple plasmids, which might seem difficult to optimize, but it's actually just a couple experiments with MaxCyte testing different ratios and concentrations, and this group was able to get a pretty good signal, as you can see here with the purple peak, using our transfection system. And so next is how we load the electroporation cuvettes, and also how the different varieties of that we have. And so we like to call it seamless scalability, which means that from the lower end with the very left side, you can electroporate with 20 to 25 microliters in total. And then you don't have to re-optimize or redo all the experiments you did to get the results you wanted at the small scale. Once you want to move over and test it with your actual samples on the large scale, say at 400 microliters or 1 mil, you don't have to redo and reselect everything. Which is quite nice. So these are all the static electroporation cuvettes, which these are all compatible on all of your instruments. So even on the smallest instrument you can do up to 3.5 mils of cells. And then here at the bottom are flow electroporation cuvettes, which we call the R20K and the CL2. These can handle up to 100 mils of electroporation volume, which is billions of cells. So if you ever need something that large, for example, for a large batch of cell electroporation, this is the correct tool to use. And so this is just a quick video showing you how to load the static processing assemblies. So you just mix in an Eppendorf tube and you put it inside the cuvette, and then you put the cuvette in the instrument and select the right settings on the computer. And so it's quite straightforward. Sometimes it gets a little trickier with the smaller volumes, but oftentimes we have an FAS on site to train your team on how to use it properly. Next is flow electroporation. So this is primarily done in the hood, but in this video, we attach a syringe to the bag. We fill the bag with our cells mixed with our DNA. And then the instrument will use the pumps to fill the electroporation chamber here, electroporate the cells, and then push them into the empty bag. And so it does this repeatedly, very quickly, so that you can electroporate a large volume in a short amount of time, and get on your way. And so this is great for if you need to do a large batch of cells and you don't want to do like 10 or 20 cuvettes, you can just use one bag instead. And so this is another representation of what we mean by seamless scalability, is that if you go to the bottom and the top ranges for the cell type or for the cell number, you really can stretch it far. And so there's a lot of overlap, so you don't really there's no like gap between two different volumes or cell types or cell numbers. And so in this next example, I'll show you how this group scaled up from around 20 to 25 microliters to 400 to 1000 microliters, and they got similar data. And so in this one, they're assaying for kinase activity, and you can measure it because the fluorophore gets redistributed throughout the cell after inhibition. And so basically, this is just the output reading, and you can see that for the small scale in blue and the large scale in red, that they all look very similar, including similar to a stable cell line, whereas this is transient transfection. All look very similar on at different concentrations. And so this shows you some scalability with our instrument. And then another example here, one group scaled up from 200 to 400 microliters to 10 to 100 mils. And in this experiment, as you can see with GPCR screening, they're measuring CCamp activity at different ligand concentrations. And no matter which scale they use, just 40 million cells or 1 billion cells, and no matter which concentration they seeded the cells at after electroporation, they got very, very similar results on both sides. And this really speaks to the consistency across dozens of across many different electroporations. And then the last step is to apply your cells to the assay. And so for that, there's a lot of data which I've organized here, as well as in this later half of the presentation. But if you'd like to read something that speaks to you specifically, we have a couple of case studies based on publications that you can read at our website, maxcyte.com/assay-ready. All right, and we've arrived at the second part, which is a case study showing you expressing difficult to produce ion channels for high throughput characterization. And so this reference paper is from Vanoye et al., and they published this in JCI Insight in 2022. So if you'd like to read more in depth, some of you might be more experts in this field than I am. Please take a read. And so their goal was to study and characterize KCNQ2, KCNQ3 ion channels, and also develop and validate a high throughput method for evaluating these variants, because a lot of the literature used manual patch clamps or did smaller scale experiments, and it was kind of inconsistent sometimes between studies. And so in this approach, they decided to generate cells for each mutant or variant that they were interested in using MaxCyte electroporation, and then they use an automated patch clamp in order to do high throughput screening. And so I think this is quite useful once you get to the data, because you'll notice that they screened close to 100 different mutants, and that would just be very much impossible to do with stable cell line generation. And so a little bit of background on this ion channel. So it's a potassium ion channel, and there's some variants of it associated with different forms of epilepsy. But because of the heterogeneity in many different types of epilepsies, it's been difficult to study. Another wrench in the plan is that there's the two different types, KCNQ2 and KCNQ3, can form different channels. And so KCNQ2 four of the subunits can come together in a homotetramer to make one channel. But more interestingly is that the KCNQ2 and KCNQ3 channel is formed by two of each for a heterotetramer. And these are where things might get interesting for the root cause for function and failure in these variants of epilepsy. And so there are many pathogenic variations, and they're dominant negative mostly. So that means they only really show up clinically as heterozygous mutants. And so it's really difficult to study because if you try to make a cell line, you can't adjust the ratios too well. And so this was kind of something that they wanted to overcome using MaxCyte electroporation. And so this is after much optimization, this is the workflow they developed. And so they have CHO cells that are already stably expressing KC and Q3, and they transfect these cells with KC and Q2 plasmid using MaxCyte. After electroporation, they treat the cells with DNase. The reason for this is that sometimes DNA-induced toxicity kills a lot of the cells, so you have to add some DNase after electroporation. And this is something that us MaxCyte FAS have lots of expertise and can show you how or when you should apply these to your experiments. After that, they cultured the cells for 2 days, and then they were actually able to use the cells fresh for the experiments or also freeze them down with liquid nitrogen and then use them later. And so whether it's fresh or frozen, if it's frozen, they let it thaw for 10 hours to recover the cells, and then they incubate the cells at 28C, which supposedly helps them express ion channels better. And then they're able to plate them, plate them for the automated patch clamp assay on a synchro patch. And so since this method is quite elaborate, they have to validate some of the points first. Now, the first point is like whether the electroporation transfection actually worked. So the CHO Q3 cells, so stably expressing KC and Q3, the ones that did not get electroporated had no signal on the synchro patch, which makes sense because the KC and Q3 subunits on their own didn't make a channel. In the second graph, the CHO Q3 cells were electroporated with KC and Q2 wild type. And so those are able to make the heterotetramer here, as you can see, which made a functioning channel, which gave them signal. Another thing they tested was comparing the automated patch clamp to a manual patch clamp, and they're able to get about the same current patterns here. And so their whole cell current density were about the same, so that helped support that the automated patch clamp was working as expected. Next, they used some inhibitors to test the functionality of the ion channel. And so here they use tetraethylammonium, or TEA, which is a potassium ion channel blocker. Different channels have different responses to this. However, they got an IC50 of around 10.7 millimolar, which matches literature values. So they're able to validate that, yes, it was transfected as expected and behaving as expected. Another drug they used is retigavine, which is a casein Q2 potassium channel opener. And so it's actually potentially used for casein Q2 associated epilepsy. So they'll use it later on in the study to test sensitivities of the variants to this drug. But typically for the wild type, that they usually see a response. And so here they saw a 1.4-fold increase in peak current density at plus 40 millivolts, which matches what they expected in previous reports. So all this is to say that they're able to validate their system and they're good to go with trying out some of the variants. And so this graph is a little bit more difficult to read, so I'll walk you through it. On the left side, I'm just showing you what plasmids the cells received. And so on the very top, the wild type cells only receive the casein Q2 wild type plasmid, just giving you a baseline for all of these values. So on the right side, all of these, for example, the current density is calculated by percent compared to wild type. The other graphs here, such as R198Q and et cetera, are all different casein Q2 point mutations or casein Q2 variants. And so these were quote unquote homozygous mutations. So in a real person, they would have 2 copies of this, and usually, typically, they have 1 copy of the wild type and 1 copy of the variant, but just for testing purposes, it was just the variant in this case. And you can see that the curves look very different compared to wild type, indicating different issues with the function. For example, R198Q you can see that the current density is slightly higher / about the same as wild type, but the midpoint of activation voltage was higher than before, and so this indicates hyperpolarizing effect of the mutation. In contrast, R207W shows a little slightly lower current density and also much lower midpoint of activation voltage. So this is showing you that the mutation is slightly depolarizing. Lastly, T274M shows you a loss of function here where the current density just is not existent and they can't measure the midpoint of activation because it didn't actually reach the peak. And so these are just a couple of examples that we pulled from the paper. And so they tested 18 variants in total. And the color coding here is that the blue is benign familial neonatal epilepsy, and then the red is early life epilepsy and developmental epileptic encephalopathy. So it's a mouthful, but it's just differentiated between diseases. Importantly, the black dots here are actually the literature values. And so if you look very carefully, 16 out of the 18 variants match and are consistent with the published values, which is quite good in terms of just validating that, yes, this method works and it matches what was previously published, so we can trust the outcomes and the data from this. And so overall, the authors are very happy with how it validated their method using this MaxCyte electroporation process as well as some automated patch clamp. And so now that we were able to match literature values, we can now look into variants that aren't as well studied. And so in these graphs, they tested around 39 different epilepsy-associated variants, but these haven't been investigated yet in literature. And they also wanted to evaluate it both in the quote-unquote homozygous and heterozygous configurations, where homozygous just means that they electroporated with only the variant, so there's no copy of wild type. Whereas heterozygous, they're electroporated at a 1-to-1 plasmid ratio of the wild type and the variant, thereby mimicking heterozygosity as seen in clinically relevant settings. And so I'm just showing you the graph for the heterozygous because it's more interesting. On the homozygous, a lot of the dominant negatives just have no measurable current density. So it's not as easy. It's a little bit like looking at a graph with a bunch of 0%. And so this one, you can see some slight shifts because with one copy of wild type, there is some function, but still a decent loss of function. And so they were able to identify several dominant negative variants where the peak current density was under half of wild type, which is kind of what you expect since only half of it will be working because half of it is wild type. And then a lot of the variants also had significant shifts in the midpoint of activation. And so we can go over the nitty-gritty details of all these variants, but you can just see here that each one of these would have been a stable cell line to somehow produce at these 1-to-1 ratios, but that is just very, very difficult to produce that many different stable cell lines manually. And so this kind of shows you how MaxCyte plus automated patch clamp workflow helps you save time and lets you do these larger studies more economically. Lastly, in this piece of data, they test these different variants' response to reticketabine. And so testing this treatment could potentially help inform whether like, oh, with this patient, with this particular mutation, this drug does or doesn't produce a response. It should or shouldn't be recommended. And so studies like this kind of can continue in the future in order to validate whether these drugs are effective against these variants. And so it's a heat map. So red means not as if was lower, and blue means higher than negative control. Without reticketabine. And then the green and the yellow also show you with reticketabine the responses of all these different variants. So in summary, if they use stable cell lines, it would have been difficult to balance the different expressions of the different subunits, for example, the wild type and the variant, it's kind of hard to balance that after you start expressing it stably in the cells because you don't know how many copies they took up. Another issue with cell lines is risk of genetic drift and some variability with each time you generate it, it could be different. And then this is also very resource and time intensive because it takes several weeks to establish a cell line. In contrast with MaxCyte transfection, we were able to dial in the plasmid concentrations fairly easily by just adjusting the ratio of one to another that they're given to the cells in electroporation. They're able to get consistent results even with cryopreserved cells, speaking to the gentleness of MaxCyte electroporation. Lastly, they were able to generate the ion channel expressing cells within days instead of weeks, and this enabled them to do the studies with the automated patch clamp and matching that high throughput method with another high throughput method. And so if you are performing studies like these, we have a lot of different customers also doing ion channel studies using MaxCyte electroporation with a mix of manual or automated patch clamps. So if this speaks to you, let us know, and we're happy to set up a conversation later to talk through about your specific needs. In this last section, I'll talk about how we can optimize MaxCyte electroporation in ion channel and GPCR experiments, and I'll show you lots of examples. And so I just wanted to share maybe some secrets, which is our key optimization parameters. And so the biggest two that I can think of is DNA concentration as well as cell culture conditions. What I mean by that is a lot of different things, but primarily what we are talking about is seeding density, like how many cells you plate per square centimeter after electroporation. Temperature shift, so the temperature you incubate the cells doesn't necessarily need to be 37 degrees. You can change it a little bit. How you freeze the cells afterwards and thaw them can also play a role in whether they are consistently able to produce results after electroporation after you thaw them, as well as some more basic things like what passage number they're at. Low passage number is better, and the phase growth they're in. We prefer log phase growth before electroporation because a happy cell in gives you happy cells out. Right, in this first example of many, I'll show you how DNA concentration and culture conditions were two variables that they optimized here. And so in the violet and then the darker purple, there are two different DNA concentrations at 50 to 150 micrograms per mil, as well as on the bottom are four different culturing methods, including 37 degrees for 24 hours after electroporation, 37 degrees for 48 hours after electroporation, and then some temperature shifts with half and half, or like 1/4, 1/3, 2/3. I think that's 1/4, 3/4 actually, at 37 and 28 degrees. And so here they're quantifying the number of seals, so how many successful measurements they got from their patch clamp. And they're able to see across all of these conditions that they're still able to get 87% or higher, which is great because that means it's saving you, it's more efficient with a patch clamp readout, so you're not wasting wells. And then on the left side, you can see that they got the highest percent of cells expressing high current at around 90-something%, with the highest concentration of DNA of around 150 micrograms per mil, with the temperature shift in 2 days. So you can see, you can think about all the other variables you could be playing with here, but I think they were quite happy with getting around 95% here. In this ion channel example, they also tried titrating different DNA concentrations. And here you can see just the plate where anything that's white means they didn't measure anything current-wise. Anything with yellow means it was a successful measurement. Anything in red means it was a measurement, but the resistance was too low, so it was excluded. And then also blue is excluded. So more yellow is good. As you can see here with no transfection or mock transfection, since the cells did not get the ion channels, they will not produce any current, which is kind of what they saw. And then they tried 50 micrograms per mil or 200 micrograms per mil of cDNA. And you can see that there's more yellow boxes for the 200 micrograms per mil cDNA, indicating that a larger number of cells were, or a larger percentage of cells were able to be measured. And so this is more efficient in terms of how much time and money you're burning on your patch clamp. And so you can see that concentration might not, it might not be the first thing you think about for like how many cells you can get, but it makes sense because the more DNA you put in, the more channels you get, the more likely the cells received the DNA and are expressing the channels, and therefore more likely you're getting signal. And here's yet another example for DNA concentration, but for GPCR. And so in this example, they used 293H cells and they electroporated them with plasmid encoding M1 muscarinic receptor. And so here they electroporated at 4 different plasmid concentrations, 0, 50, 100, and 200 micrograms per mil. And then afterwards, they treated it with carbachol and measured calcium flux via a fluorescent assay. And so you can see here that the lower concentrations in a darker color, they got less response from it, which means that probably less of that M1 receptor was expressed. And then with increasing DNA concentrations, they were able to get higher response signal. And so maybe they could titrate it higher than 200 micrograms per mil. But this just shows you how the DNA concentration can really help you fine-tune your assay. Here's another example for DNA concentration with ion channels. In this case, we have CHO-K1 cells, and we transfected them with a plasmid encoding KB1.3 GFP. And so this is an ion channel with GFP attached, and you can see that the cells were clearly turned green, which is a good sign. And then at 4 different concentrations, including 0, that they are able to titrate to see which produced the most current amplitude. And surprisingly, more is not always better. So 80 micrograms per mil here of DNA worked the best for them. And then in this other example from the same group, they did a different channel, KV 1.5, and you can see that they titrated a different set of concentrations and got slightly different results, where around 100 and 200 micrograms per mil looked about the same, and they're able to get over 2 nanoamps of current. And so this maybe just indicates to you that like even though you titrate it once with one channel, you might want to test a titration again with another channel in case you're getting problems because it could be channel or dependent or plasmid dependent. In this example, plating density actually changes how the ion channel signal comes out. And so this hex cells were electroporated with a construct and then also seeded into a 1536 well plate. After that, they're stimulated and then you can see calcium flux. And so here you may not really expect it. It might not be super obvious to you immediately, but it did seem to change depending on the seeding density. And so with higher seeding density, they're able to get a more steep response curve. And then at lower seeding densities, they're still able to see a curve as well. And so they also compared fresh versus frozen, and they got comparable results. And then a quote from this paper is that to further minimize assay variation, pilot screens were all performed using a single batch of transfected cells prepared in bulk then frozen. And this is a very vital key point because many electroporation instruments may not preserve the cell viability as well as MaxCyte, so you can't always do this. And so with gentle electroporation with our instrument, they were able to execute this experiment and be able to do their technical replicates without any problems. Lastly, I'll talk a little bit about how you can optimize the cryopreservation part. And so this is kind of a simple general workflow on how people freeze the cells after electroporation. And so after electroporating the cells, we often ask people to plate the cells and then try 2 or 3 different time points after transfection of how long to incubate it for. For example, overnight or like 2 hours, 10 hours, just to test it. And then after that, you can lift the cells and freeze them. We have recommendations for the amount of DMSO to put in, but it can vary depending on the cell type and your setup. And so you should try different variations and consult us for suggestions. After that, you can incubate the cells a little bit immediate before you can plate them at different times before freezing, and also try different ways to thaw them because it can vary depending on the cell type and the experiment. This is an example using a reporter assay for in Jurkat cells. So here you can see that the fresher cells and the cryopreserved cells look very similar in terms of their IC50, despite the cryopreserved cells being frozen and then thawed, which speaks to like some good optimization on their part for the electroporation. In this example with ion channels, you can see that they cryopreserved the cells and saw that very little change or drop in performance. And so these CHO-K1 cells are transfected with KV 1.5 plasmid, and then their cells were cultured for 37 degrees for a day, and then cultured at 28C for another day, and then they were cryopreserved. And so you can see that here, the ones in August, they got around the same percent sealed for the different plates here. And this is just to summarize in case you want to screenshot it or talk to us about these points, is that we do have lots of experience in ion channel and GPCR experiments. So if you're struggling with consistency or want to try the MaxCyte platform, we're more than happy to talk to you about it. And so that wraps up today's talk. So today we talked about introduction to MaxCyte electroporation and specific examples for how it works for ion channel and GPCR assays. I spoke about a case study with ion channels for high throughput patch clamp assays using MaxCyte. And then lastly, we talked about how to optimize MaxCyte electroporation for ion channel and GPCR experiments, including many examples of how other groups have optimized it and applied it to their studies. So thank you so much for attending today's talk. I'm live now for Q&A. If you'd like to ask any questions, please feel free to put them in the chat. Thank you.